Type 2 diabetes is a metabolic disorder characterised by an inability to utilise insulin effectively, as well as a reduced capacity to produce insulin. This results in abnormally elevated plasma glucose levels. Type 2 diabetes is currently considered the fastest growing chronic condition in Australia. There are approximately 1.2 million Australians currently living with diabetes, of which 85 to 90% are type 2. Over the past 25 years, the prevalence of diabetes has tripled. The rate of diabetes is similar in major cities as it is in regional and remote areas. However, in regions of low socioeconomic status, the rate of diabetes is almost double. Approximately 13% of Indigenous Australian adults have diabetes. It is estimated that Indigenous Australians are almost 4 times more likely to develop diabetes than non-Indigenous Australians, and many tend to develop the condition at a younger age. Type 2 diabetes most commonly develops in individuals who are 45 years and older. The age demographic with the highest number of reported cases is 75 years and above. However, there is an increasing number of children, adolescents, and young adults who are being affected. The exact cause of type 2 diabetes is unknown. However, it is generally understood that it is a multifactorial disease that arises due to several contributing factors, including genetic makeup, lifestyle, diet and eating habits, and environment. There are several genetic variations associated with type 2 diabetes that act by altering gene expression. Changes in gene activity may be responsible for dysfunction of the pancreatic beta cells that produce insulin. They may also affect the processing and recognition of insulin and cellular sensitivity to insulin. While some individuals may have a stronger genetic predisposition towards type 2 diabetes, it is likely that these genetic variations act in conjunction with lifestyle and environmental factors to promote the development of diabetes. For example, an individual who consumes a diet high in fats and sugars and low in dietary fibre and nutrients, combined with a lack of regular physical activity, is at a high risk of becoming overweight or obese. People who are in this category are at a higher risk of developing type 2 diabetes. This is because they tend to have lower muscle mass and increased lipid stores, which can cause insulin resistance. There is strong evidence to support that if genetic variation and a poor diet and lifestyle are combined, insulin resistance and impaired insulin secretion can develop. In normal physiology, insulin is a hormone secreted from the beta cells of the pancreas. It has several important biological functions that all work to integrate body fuel metabolism. In a feeding or fed state, insulin is secreted into the bloodstream. Here it is responsible for decreasing the mobilization of endogenous fuel stores, stimulating the uptake of carbohydrates, amino acids, and lipids into target tissues, and replenishing fuel stores that may have been depleted during periods of fasting. Insulin is therefore critical in the tight regulation of plasma glucose concentration. Ingestion of food triggers a series of signals to several body tissues that subsequently initiate enteric factors to be released, including cholecystokinin, glucagon-like peptide 1, and gastric inhibitory polypeptide. These enteric factors then trigger the pancreas to enhance insulin secretion. When insulin is secreted, it binds to a tyrosine kinase receptor in the plasma membrane. This then causes an intracellular signal cascade that results in the recruitment of vesicular-bound GLUT4 transporters to the plasma membrane. The recruitment and activation of these transporters stimulates the movement of glucose into the cell, effectively lowering plasma glucose. In type 2 diabetes, this signalling pathway is impaired. Insulin still binds to its receptor, but the downstream signalling cascade is disrupted, meaning there is no recruitment of the insulin-dependent GLUT4 transporters to the plasma membrane. As a result, there is a decrease in glucose influx and plasma concentration remains high.
The first thing I noticed was that I was incredibly thirsty all the time. Didn't matter how much water I drank, I was never able to feel like I'd had enough. And I was also very fatigued. I was getting a good night's sleep, but regardless, I was tired all day long. And the thing that finally forced me to go to the doctor was I was getting pins and needles in my fingers and toes.
One of the most common symptoms of type 2 diabetes is feeling tired. This is because there is not enough glucose getting into the cell to enter ATP-making pathways, so energy levels are low. Other signs and symptoms include excessive thirst, excessive urination, slow healing wounds, infections, and tingling sensations in the hands and feet. However, often people who have type 2 diabetes remain asymptomatic for several years, so can be living with diabetes undiagnosed. Prolonged exposure to high plasma glucose levels can cause significant damage to other parts of the body, increasing the risk of developing cardiovascular disease, peripheral neuropathy, renal damage, hearing and eye damage, and sleep apnea. Type 2 diabetes can be a difficult condition to manage. The main goals in the management of diabetes are to eliminate many of the associated symptoms to prevent or slow the progression of further complications. Lifestyle, diet, and exercise habits are largely modifiable. By adhering to a healthy diet, losing any extra weight, and exercising regularly, it has been shown to reduce the severity of diabetic symptoms. However, there is no known cure for the condition, and therefore prevention mechanisms are vital to slowing the rapidly developing rate of type 2 diabetes.